Hello, today you are going to watch me virtually dissect a mushroom. So this is a white mushroom. It's the common type of mushroom that you find on salad bars or when you order mushrooms on your pizza. It is in the phylum Basidiomycota. Remember, Basidiomycota, all members of that phylum have a structure called a basidia, which is a spore-producing cell. It's shaped like a club. That's why they're also called the club fungi. All right, so first we're going to look at the different parts of this mushroom. Then we are going to use a microscope to look for the basidia, and we're also going to look for the microscopic little hair-like hyphae. So what you need to do is follow along on your worksheet, and you're going to be filling out those questions based on what you see in this video. All right, so first, basic parts of a mushroom. They're not super complicated. All right, remember that the mushroom is not the main part of the fungus. When you think of fungi, you usually think of mushrooms because that's the part that we see. Most of the time, most of the fungus, is underground. It's the mycelium. Remember that big network of hyphae that travels all over underground. One mycelium might produce multiple mushrooms. Remember that the mushroom is just the fruiting body of the fungus. It's the structure that comes out of the ground, produces spores, and then facilitates the spores' entry into the environment. So, basic parts of their mushroom. The big top part is called the cap. The part connecting the cap to the mycelium is called the stalk. Right, this mushroom is obviously cut at the bottom for harvesting, but if you just plucked it out of the ground, you would see some little bits of the mycelium, sort of like little roots almost, coming off of the bottom. All right, so the first thing I'm going to do is I'm going to crack the stalk off of the mushroom. All right, got that removed. We're going to come back to that in just a second. We're going to look on the inside of the mushroom. All right, this little protective coating that sort of connected the stalk to the cap is called the veil. We're going to go ahead and peel back the veil. And underneath, you'll see the important part, which are called the gills. They look sort of like fish gills, and there's all these little slits. Now, covering the gills are all the spore-producing basidia. So what we're going to do now is we're going to go ahead and crack open this stalk, try to isolate some hyphae to look at. Because remember, all parts of a fungus are made of hyphae, even the cap and the stalk. And then we're going to pull out a gill, put it on a wet mount slide, and we're going to look at it under the microscope along with the hyphae as well. All right. So first up, got my cap, my stalk. I've got some slides and some cover slips and some water to make a wet mount slide, just like we've been doing all year long. Go ahead and crack this stalk open. You should see lots and lots of little thread-like projections in there. Those are all of the hyphae. Many of them bunched together, sort of like if you undid a rope, there'd be lots and lots of little pieces of twine. Sort of a similar concept. A whole bunch of them come together to make up the fruiting body. All right, so we're going to look at some of those under a microscope. I'm going to prepare a wet mount slide, and I'm going to go ahead and break this cap in half. I'm going to use some forceps to go ahead and pull a gill out. We're going to look at one of the gills under the microscope, and all along the gills we should see the little club-shaped basidia, and if this is a mature mushroom, we'll see some spores in there too. All right, so what you're now seeing is my screen on my computer. I hooked up one of the fancy camera microscopes to my computer and put a piece of the mushroom stalk underneath it. Remember, we used these camera microscopes for one of our microscope labs this year. So if you look at the stalk very closely, I'm going to use my cursor to show you. You can sort of see this wave-like pattern. And remember how I said that the whole stalk was kind of like a big cord or a big rope, And then if you looked at each little fiber, each thread, there were a bunch of little pieces inside of it, of threads inside of it. And then you look at that thread and there's more threads inside of it, just like how we build a rope. We combine little threads over and over and over and over again, and eventually we get something strong. So this is a piece of the stem under 40 times magnification, so under the lowest power. But if I then take my scalpel that I use to slice the stem with, the knife, and wipe it on the slide, I'm able to see some of the individual hyphae. So these are all the way down the smallest unit of that little rope, the smallest pieces of fiber, the individual hyphae. And they're actually pretty beautiful in these little kind of star-like patterns. So I'm going to find some that I'm going to zoom in on. We'll look at them a little bit closer. All right, so I'm at 40x, 40 times magnification. I'm going to zoom into 100 times magnification. You can see all of the individual hyphae. Remember, the hyphae make up, are the basic unit of a fungus. They make up everything in the fungus. The fruiting body, the mycelium, all of it. I don't know what my camera's doing right now. It's spazzing out. All right. Next, I'm going to show you the basidia, the spore-producing structures on the gills of the mushroom. All right, so now what you're looking at is one single gill that I pulled out from the cap of the mushroom. And right along the edge of the gill here, this is the edge that would be exposed to the air, so pointing down back towards the ground, you can see the little basidia sticking off. We're going to zoom in just a second. This is at 40 times magnification. Here's the gill, and on the edge are all the little basidia. Remember, the basidia are cells that produce spores externally, so not on the inside of the cell, but on the outside of the cell. And they're club-shaped. They're the reason that these fungi are called club fungi, and also the namesake for the phylum Basidiomycota. All right, we're going to zoom in a little bit and see if we can find some basidia up closer and then some spores as well. All right, so on to 100 times magnification. Here we go. We've got some good basidia. You can still see them, those little finger-like projections. I'm just going to try and center them before I zoom in one more time. All right. Let's get a little bit better focused. Here we go. 400 times magnification. All right, you can see the club-shaped basidia. I believe there's a little spore hanging off of one of them. See if I can get it in a little bit better focus for you. And there are tons of them. They literally cover this entire gill. They're all over the place. I mean, just right here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 in this little tiny, tiny spot. So remember, one mushroom can make billions to trillions of spores, tons and tons of spores coming out of this thing. All right, and I actually, when I put my cover slip on, I released a little cloud of spores that we're about to go try and find. So we're zooming back out to 100 times magnification. There's our cloud of spores that sort of shot out when I put the cover slip on. Let's go ahead and center that. All right, let's zoom in on them and see if we can see some spores and see what they look like. Okay, so still at 100 times magnification, going up to 400 times magnification. There are some individual spores. Pretty cool. Okay, so quick review. Make sure you know the cap, the stalk, the gills, where the basidia are located, and also where the spores are produced. All right, make sure you're filling out the questions that go along with this video and uploading them to my Berkeley.